Today we're going to be talking about Watson and Crick's DNA model. Right here I have an example of a right-handed DNA. As you can tell by the top of it's pointing towards the right and it moves towards the right. And that's going to be the structure of normal DNA, because right-handed DNA is more structurally sound than left-handed DNA. And right here we can see I have Twizzlers representing the phosphate backbone and these toothpicks representing the base pairs. So with our base pairs, I'm going to have blue is for adenine, red is for thymine, yellow is for cytosine, and black is for guanine. And adenine and guanine are going to be purines because they have a 2-carbon ring structure, and cytosine and thymine are going to be pyridamines because they have a 1-carbon ring structure. And the bases are stacked alternating, so we have thymine first, then guanine, then adenine, then cytosine. And these bases are held together by hydrogen bonds. And guanine and cytosine are going to have 3 hydrogen bonds that hold them together. And adenine and thymine are going to have 2 hydrogen bonds that are holding them together. Okay. And if we want to look closer on a nucleic acid, we can see that they're made up of ribose sugars right here, and we have first carbon on this side, second carbon, third carbon, fourth carbon, and the 5th carbon is not attached to the ring. The first carbon is important because it bonds the nucleic acid bases, and the 5th and 3rd carbons are important because they connect to the phosphate group. So between the backbone, we're going to have minor grooves and major grooves. Right here is going to be a major groove because there's a big space between the backbones, and this is important because it allows for proteins to be able to bind to the DNA. And then we have minor grooves shown like right here where the backbones are extremely close together and no protein is going to be able to bind there. DNA is anti-parallel, meaning that if we have a 5' end here and a 3' end here, then we're going to have the opposite on the other side, the 3' end up here and the 5' end down here. And this is important because it creates a leading and a lagging strand, which are the continuous and non-continuous parts of DNA replication, which is what makes replication possible. So DNA backbone itself is going to be negative, but if we were to have sodium come in, and sodium is positive, it would neutralize the DNA and make it more positive, which is going to make it hydrophobic and increase its melting temperature because it's going to be harder for these bonds to break apart in water if it is hydrophobic. And here are my references.